I'm Salma Quraishi. Welcome to Neuroscientists Talk Shop, the University of Texas at San Antonio's neuroscience research podcast. It's October 29th, 2020, and today we welcome surgeon scientist Philip Starr. Hi, Philip. Can we hear him? Oh. Hello?
Hello.
Oh, I'm sorry. We were doing the voice stamp. I should have explained that because this does end up as audio. Okay, that's fine. We'll have a little glitch there, but that's okay. Philip holds the Dolores Cakebread Endowed Chair in Neurologic Surgery and is a faculty member of the UCSF graduate program in neuroscience. He co-directs the UCSF Surgical Movement Disorders Clinic, which is one of 4 national Bachman-Strauss Centers of Excellence in the treatment of movement disorders. His clinical interests are in functional neurosurgery, specifically in the use of implanted devices to improve brain function. His research program, his very active research program, works in tandem to decipher how movement disorders alter brain network function, also to understand the mechanisms through which neurostimulation remediates symptoms, and finally how to refine and build better, more mechanistically informed stimulation protocols and devices. Such a pleasure to have you. So in the gallery today, we've got 2 basal ganglia network specialists, good old Charlie Wilson. Hi, Charlie.
Hi.
And we've got Matt Higgs. Hi, Matt.
Hi.
So I want to point listeners and viewers who are interested in neuromodulation and Parkinson's disease to check out our 2019 Brain Oscillations in Parkinson's Disease Symposium podcast, which lives in our audio archive and has Charlie leading a really fun discussion with Philip's colleagues, Jerry Vitek, Judy Walters, Rob Turner, and Mark Bevan. So let's get started. So Philip, you work on many areas in motor disorder research and in stimulation-based therapeutics. And just one of these includes identifying neural signatures in Parkinson's patients as a first step in programming a new generation of smart devices that you're currently one of the people really at the leading edge of. And these monitor the state of patient brain networks concurrent with movement biometrics to deliver precision stimulation as needed. So it sounds like science fiction, but it's here. So the clinical relevance here it prompts so many questions that we could keep you for hours on that topic alone. But given that our discussions typically center on questions like, what do the basal ganglia do? I thought a good starting point might be to talk about this goldmine of individual human field potential recordings that you've got from motor cortex simultaneous with one of 2 different basal ganglia nuclei, which were then cross-coded to movement biometrics. I mean, that seems huge. So clearly the priority is in a study is to understand and treat the individual patients first and foremost. But can you start us off by telling us about this really important work and how it's helping us understand oscillatory synchronizations in motor disorders and in whole brain motor systems? Just, give us your nutshell.
Sure. Well, I've been involved in deep brain stimulation for 22 years and was recruited here to UCSF to start the program in DBS in 1998. And I started out being very intrigued by how deep brain stimulation works, and I was very influenced by Malin DeLong's group, where I had some training at Emory University. And I started out, looking at single units in the basal ganglia intraoperatively, and in humans, we're only able to record individual brain cells one at a time. And of course, as it's turned out, there's more and more interest in the idea that synchronization across populations of neurons is really important in the expression of movement disorders, motor signs. And that's hard to do in humans with single units because you'd have to record dozens or hundreds of single units at once, which is possible in an animal model, but difficult in humans. So, I followed the work of Peter Brown and others in the early 2000s about what could be extracted, the incredible information that Brown and others extracted from local field potentials in humans in the subthalamic nucleus. These were tiny signals, 10 microvolts peak to peak. And I wanted to take that same approach with a multi-site recording approach. So, obviously the basal ganglia are connected to a network involving the thalamus and various parts of the cortex. And I felt that recording multiple sites simultaneously in that network would be a good way to advance understanding and take some of the analytic methods that Brown's lab and others were working on, 15 years ago, and apply that to a multi-site approach.
So the question about these pathological rhythms, so we always think about Parkinson's disease being about dopamine neurons and the depletion of dopamine neurons. And it's really clear at this point that the real issue is not the loss of dopamine, it's the pathological rhythms that are potentially, that are causing the motor disorders themselves. So can you talk about the fact that most of these networks, like, so how much of this is a compensatory issue that we're talking about versus sort of like the actual issue itself is these pathological rhythms? And then I was hoping you could also say something about the fact that we have so little access now, it seems, to untreated brain. So levodopa is still the current therapeutic that everyone relies on, right? And that itself is producing potentially so many cellular changes in this network that's then reinforcing these oscillations. So I guess my real question is about that, right? Is about how this is an old technology. We've been stimulating brains for a while, and the levodopa has only been around since the 60s. Are there any clues in some of the untreated, are there any untreated recordings that give us any sense of what untreated pathophysiology looks like versus levodopa pathophysiology? And then, of course, there's a compensatory issue question itself, right? So there's a lot in there.
Yeah, no, you raised the excellent point that a levodopa treated brain is different, right? And even if you wait, hours or days for the levodopa to wear out pharmacologically, to wear off, there's still long-term changes in the brain, and many people have looked at this in animal models, particularly the long-term changes in corticostriatal synapses induced by this abnormal pulsatile, release of levodopa from taking medications. It does come about that we have some patients that we treat surgically and can record from who really have not been on levodopa. Some are just quite intolerant of it for various reasons, either severe nausea, or some young onset patients really get severely dyskinetic with a minimal dose of levodopa. So I have treated people who have not been on Parkinson's medication other than to have tried it, to done a test of it. Although we don't currently have one such patient in our long-term invasive recording studies. We have had patients in the intraoperative recording studies, which is of course more numerous numbers of patients who have not been on levodopa. I think that the kind of off medication features of those brain networks seem similar. There is, a strong degree of synchronization between neuronal areas. We don't have normal controls, so I'm always careful to call something, abnormal when we don't have normal controls in invasive recording in humans to work with. But we do have other movement disorders that we treat identically. We treat, for example, isolated cervical dystonia, another movement disorder which only affects the neck, and the rest of the body is normal. And so that provides one type of control where we're in the same structures. So that's one way we get around the lack of a normal control. We also, there's some misconceptions in the field that when people started looking at subthalamic nucleus oscillations in the beta range and found that therapy, that is levodopa or DBS, suppresses oscillations in the basal ganglia in beta in proportion to the degree of therapeutic benefit, people came to the conclusion that, well, beta activity is abnormal and is a hallmark of the Parkinsonian state. And I never accepted that. Clearly, what's been well established is that beta band oscillations in basal ganglia nuclei are a good marker of effective therapy, right? They reduce as you give levodopa, therapeutic levodopa or deep brain stimulation. That doesn't mean that they were absolutely abnormal to begin with. And in fact, when we've recorded in cervical dystonia, and Hagai Bergman and others have recorded subthalamic activity in obsessive compulsive disorder, so a non-movement disorder, no dopamine loss. And we find beta band activity as would be expected as a marker of the motor system. So, so these oscillatory activities are certainly a marker of therapy. There is some emerging evidence that they also are truly exaggerated. My bias from my read on the literature is that subthalamic beta band oscillations are not necessarily vastly elevated themselves, but other aspects of beta synchronization in the motor network probably are quite exaggerated. So for example, coherent oscillations between basal ganglia and cortex are likely quite exaggerated, even though the beta band activity within the structure may not be so much. So it's possible that levodopa and deep brain stimulation reduce subthalamic beta oscillations way beyond, beyond what is normal, right? Reduce it to pathologically low levels, but that is a mechanism whereby they can disrupt synchrony between basal ganglia and cortex, which is, a state of excessive communication between those structures. I lost the other part of your question.
I mean, well, we can move on, but I think that the other thing, it was about the compensatory, whether these are, how much we can sort of think about these as compensatory versus actual, like the initial insult kind of thing, and where that leads us in terms of DBS, in terms of programming DBS and trying to improve things that are more than just about akinesia, I guess. And that's another thing I want you to talk about, is the fact that, there are multiple syndromes and symptoms that these address. But go ahead.
It's always tough in human research to know what is primary and what is compensatory, right? Because in animal models, you can do more manipulations. You can see if something is causal. You can optogenetically drive something in the way that you want, and look more at causality. And we're not so much able to do that in humans. There are certain things we suspect are compensatory. For example, we noticed in looking at movement-related oscillatory desynchronization in the beta range in Parkinson's patients compared to non-Parkinsons, like essential tremor. There's actually increased movement-related beta desynchronization. That is, when you move, you have to desynchronize your beta oscillations in order to make normal movements. And interestingly, Parkinson's patients do that more so than, say, essential tremor patients, which to me might reflect that they have an exaggerated state of beta to begin with, and therefore they must compensate for that. Obviously, Parkinson's patients can move. They don't move normally, but they can move. And so to initiate any movement, they have to really excessively damp down that beta synchrony even more than other patients. So that's an example of something that may be compensatory.
So I'd like to get your impression about what's happened to the model of basal ganglia and Parkinson's disease. So in the old rate model, it was really easy to say why Parkinson's patients aren't able to move. It's because the thalamus has been inhibited, and the thalamus is somehow carrying a signal that's required for movement to the motor cortex, although that signal wasn't well defined and its necessity wasn't clear, but it did kind of make sense by analogy to the visual system, where you can't see if the thalamus can't tell the cortex what's going on in the retina. Somehow you can't move if the thalamus can't tell the cortex what's going on in the cerebellum or something like that. So that was that. But in that idea, the thalamus was sort of shut down. And now we think there's just some kind of oscillation in the thalamus now that's coming from the basal ganglia. So how do we think that is causing a problem with, because the movement problem isn't an oscillation itself, but there is a kind of tremor, but that's not what we're talking about.
So yeah, that's a great question. And, the original rate model that started off this whole field of deep brain stimulation, did envision the basal ganglia output, the internal globus pallidus, as being an overdrive. It's an inhibitory GABAergic structure. And so it was flooding the motor thalamus with GABAergic tone and shutting it down. And then therapy, like lesioning the globus pallidus, reduced that abnormal inhibition on the thalamus and allowed it to communicate with the cortex, right? So interestingly, I think the oscillation model does provide a sort of related concept if you use the so-called communication through coherence hypothesis that, has been elucidated very well by Pascal Fries and others in the field. And that is, that structures that oscillate together talk to one another. So let's say there's a state of excessive oscillatory coherence between the globus pallidus internus and the thalamus, right? That's can be also similar to simply inhibiting the motor thalamus. And if you then disrupt this oscillatory coherence, you can, between basal ganglia and thalamus and then cortex downstream, you can interrupt this pathological state of over inhibition of the thalamus, but it's not by blocking specifically action potential discharge, GABAergic action potential discharge. It's by, dephasing, getting thalamus and basal ganglia to not oscillate together. So the same idea can apply. You can then release the thalamus and the thalamocortical pathway from this excessive effect of basal ganglia.
So maybe there's some other structure that the thalamus is trying to be coherent with, and it can't because the basal ganglia is just creating a bogus coherence. And is that what you're thinking? So you can't be coherent with two dephased inputs at the same time. If one of them grabs you, then you're stuck listening to that one.
It could be part of it, although it's also quite possible, and there's evidence that basal ganglia, thalamus, and cortex are all excessively coherent. So the way that I look at it is, you still have to consider the excitatory and inhibitory action potential discharge pathways. And so why does coherence between globus pallidus and thalamus matter? Well, it's because if they're highly coherent, those GABAergic action potentials from GPI will arrive at the thalamus at the correct phase to excite further action potential discharge, or correct phase to, in this case, be inhibited, excessively inhibited, because it's a GABAergic projection. So I think you could postulate that one structure is locked in phase with another and therefore separated from the system, but I think you don't have to. You can simply postulate that the oscillatory synchrony between basal ganglia and thalamus, if it's too great, means that action potential discharge from GPI is excessively inhibiting the thalamus. And the thalamus may still be oscillating with cortex, but it is still over inhibited because of that synchrony with globus pallidus. It's hard to describe without a lot of diagrams, but that's kind of the idea.
So that's the great explanation, I guess, for just not moving, because there's some signal from the thalamus that's required for movement. And nobody, as far as I know, we don't know what that signal is or why it's important for movement. But assuming that that's what it is, then the thalamus can't give it. But the basal ganglia has connections to a lot of non-pyramidal motor structures that alter movement. The pontine reticular formation gets a big input from the basal ganglia and the spirit colliculus. And this old idea, the oldest idea maybe about basal ganglia, is that it's controlling non-pyramidal cortical pathways rather than pyramidal ones. Do you think that the non-pyramidal globus pallidus outputs are going to have a renaissance of interest? Are they part of what we're looking at in the patient?
Yeah, it's possible. In fact, there's already been a mini renaissance of interest in the pallidal outflow to the PPN, pedunculopontine nucleus. There was some work empirically that indicated that modulation of the PPN could improve gait freezing, and that led to a little burst of clinical trials in neuromodulation of the PPN. Again, reflecting what you talked about, basal ganglia output to brainstem to spinal cord, bypassing the motor cortex. Those studies didn't work well enough to make that a mainstream effect. We did do PPN stem in a small number of patients, and there were often effects on gait for a few months. These are typically, though, disorders with relentless neurodegeneration at that stage with advanced gait freezing. So it's a hard thing to treat with just neuromodulation. But some people are still working in the background on treatments for gait freezing, with the idea that gait problems may use a lot of that pyramidal tract function.
So the differences in symptomology, though, you pointed out that that individual patients have very different signatures for how the different types of oscillations are distributed in cortex versus the basal ganglia nuclei, according to what's happening to them in their motor state. But what are the kind of ideas about why we see that, and why would DBS work for everything? And you're now seeing that in fact it doesn't work for everything. It has to be sort of titrated into very specific spatiotemporal. I mean, it seems like there is more of a temporal component. Is that true, or is there a spatial component also? Do you place probes differently depending on where you see the symptomology?
So yeah, those are good questions. There is some tendency to try to place therapeutic electrodes differently depending on symptomatology. For example, in both the internal pallidum and the subthalamic nucleus, for patients with very strong tremor, or so-called tremor-dominant Parkinson's, the anti-tremor effect is a little more posterior than the anti-akinesia effect. And I've repositioned brain leads in patients who you maybe have a posterior lead, good anti-tremor effect, not an anti-akinesia effect, and vice versa. So there are these subterritories. And my bias in subthalamic nucleus is that, we know from non-human primate work that within the motor territory, the subthalamic nucleus, there are multiple functionalities. There's what you could call an M1 receiving area, subthalamic nucleus closely related to primary motor cortex. There's an SMA receiving area, that part which is slightly more anterior within the motor area, closely related to the SMA. My own guess is that the M1 receiving area, very posterior, is more involved in tremor, or that circuit can be modulated for better tremor therapy. And the SMA receiving area, slightly more anterior within the motor territory, is more involved with the akinesia. You raised another question, though, is why is Parkinson's so variable? What determines whether a patient becomes rigid akinetic with minimal tremor? What determines if they become tremor dominant? It's very unclear how this happens. And then there's other variations. We have patients with genetic mutations. We have a patient in our long-term recording studies with the Parkin mutation, a very young onset patient, and that may still have yet other patterns. There's still a lot to figure out about why there are so many differences between different patients and what the underlying pathophysiology is. Tremor, of course, is very different from the other Parkinson's symptoms in that it doesn't progress in time. So, most Parkinsonian signs, slowness, rigidity, gait disorder, postural instability, they will not get better. They will tend to get worse over time. Tremor is an odd one. You can start with tremor, and then it can actually improve as the overall disease is worsening. So it doesn't seem to correlate with degree of pathology that is dopamine loss, the way the other symptoms do. Could tremor be some kind of compensatory effect for other problems in the circuit? It's not clear.
So subthalamic nucleus has a bunch of regions. Only some of them are motor. Some of them are non-motor. I guess you probably try to stay away from those regions. But what happens when the stimulation goes into the non-motor regions of subthalamic nucleus?
That certainly happens. The subthalamic nucleus is about the size of a peanut, and the electrode, the closest spacing we have still has an array that's about 7.5 millimeters long. So you're always going to have contacts that are in the non-motor area. The non-motor area itself is about 4 millimeters long, along the tract of the DBS lead, and so you have a 7.5 millimeter array, and you're going to be able to stimulate other structures. There's a whole wealth of studies on ventral STM physiology and stimulation based on the fact that most of us deliberately overshoot the motor territory with the lowest contact and put it deliberately in the ventral STN, which is more associative and cognitive, it's thought, in the trajectory that we take as opposed to the limbic area. So we have contacts there. Some of them are stimulated either deliberately or inadvertently. There are certainly case reports of people getting worse mood depression from the ventral STN stimulation. There's varying effects of cognition. There's some studies indicating increased impulsivity from ventral STN stim. Others indicating maybe you can treat impulsivity. So it's tough to figure out. One of my only formal studies in the ventral part of the STN was looking at the physiology of stopping. So there's a lot of interest in a prefrontal subthalamic pathway, maybe being involved in inhibiting motor action, cognitive control of stopping, basically. And there is a hyperdirect pathway from inferior frontal gyrus on the right to the subthalamic nucleus via a hyperdirect monosynaptic projection. And that the physiology of that pathway can predict the stopping speed of a person. I had a graduate student, Whitney Chen, who did some nice work on that. That's the only work that I've done personally in ventral STN. It would be, there's people who have electrodes in limbic STN, right? The limbic area is a little more anteromedial. I don't usually go through that. Most people don't go through that area on the standard approach to the motor STN, but limbic STN has been implanted more or less deliberately in OCD, for example. And so there's a lot of opportunity to study that. And there's studies showing it's pretty effective for OCD.
It's amazing a little peanut-sized place in the brain would have so many different things in it, and that they would all fit. And then it would turn out to be the hub for a lot of treatment of a lot of different things.
Yeah, and people often ask, especially because I do a lot of studies with motor cortex leads, and say, well, can't you stimulate the cortex? And maybe you don't have to go through the brain. Maybe you can use a cortical lead to stimulate. And my guess is if the problem of akinesia, say, is from an abnormal oscillatory connectivity throughout the network, sure, you should be able to intervene anywhere in theory to interrupt that. And the fact that pallidal and subthalamic stimulation work equally well for Parkinson's and many disorders does back up the idea that you can go at multiple places in the motor network to interrupt the abnormal activity. Why not the cortex? I bet that if you could cover a whole lot of the cortex, motor or supplementary cortex, you should be able to affect pathological activity. I think the uniqueness of subthalamic nucleus especially, and also globus pallidus, is how the convergence, the incredible convergence of multiple widespread brain areas on those targets that make them quite effective. Whereas in the cortex, if you wanted to effectively modulate the same territory, you'd have to have a gigantic electrode, folding into the sulci, et cetera. So it's generally been a theme in DBS and even the new indications in OCD, in depression, in various pain syndromes, that you get more mileage from these subcortical nuclei where there's enormous convergence. Doesn't necessarily mean that is the site of the where the pathological activity exists, right? It's very likely a delocalized network phenomenon.
So what special? Oh, sorry. Go ahead, Matt.
Yeah, I had sort of a general question about the DBS. You talk a lot about interfering with pathological patterns of activity, sort of like maybe a soft lesion. But is that all you're doing, or is this somehow enabling normal signaling to take place that wasn't happening in Parkinson's?
Good question. So you're right. We do talk in this field often about suppressing the abnormal activity. Are we allowing more normal patterns of activity to occur? Certainly the idea is by removing this excessive synchronization that you will allow more normal activity. So for example, it's thought that to do a normal movement, you need to transiently synchronize functionally related areas and do so in a very task-specific way. So in the Parkinsonian state, or this is probably true in dystonia as well, when you overactivate neighboring muscle groups, you lock these circuits into an inflexible pattern where they cannot transiently synchronize and desynchronize. So when you release the network from this excessive synchronization, then perhaps that transient synchronization, which is a part of normal brain function, is allowed to occur.
So what's special about 100 hertz? So that's the general frequency. It's just the amplitude that you modulate and the location of the probes. Is that right?
Yes. In general, 100 to 200 hertz. Really, that came from, it started with the early studies of Benabide in Grenoble, France, on thalamic stimulation and tremor in the late 80s, where he did a frequency titration, looked at effects on tremor from thalamic stim as a function of frequency, and you had to get over about 100 hertz to have a beneficial effect. And that's been applied to Parkinson's. That being said, there are subtle differences and some therapeutic implications of changing frequency. I think the general answer is, why do we need over 100 hertz? The hand-waving answer to this is the pathological rhythms you're trying to disrupt are all lower frequency, beta, theta, alpha, to some extent gamma. And so if you drive the system or you add a stimulus at a very different frequency, you can disrupt those the pathological frequencies. Now, an obvious question is, well, what if you did 20 hertz stimulation and tried to drive the motor system at 20 hertz? Could you increase bradykinesia? That would be doing causality, right? No longer just a correlation of beta with bradykinesia. Let's drive it and let's see if we can cause bradykinesia. Peter Brown and others have shown this. It's quite difficult to show, but you can get subtle worsening in bradykinesia from 20 hertz stimulation. It probably, to really show this effectively, you have to give phase locked stimulation, right? So it's just like pushing a kid on a swing set, right? You will increase the oscillation if you deliver your energy at the right phase. You'll decrease the oscillation if you deliver it at the right frequency, but wrong phase, right? When they're moving the wrong way. Same thing with stimulation. We don't yet have the technology to do phase locked stimulation from an implanted device, but those groups that are starting to do it with fancy externalized equipment with externalized leads for using phase locked stimulation can start to look at these questions of, can we causally drive an abnormal motor sign from driving the system at what we think are these pathological frequencies. Now, I also, in response to your question, wanted to mention that people are now exploring changes in frequencies to address different symptoms. There's an observation, if we program someone at 130 hertz and they have a lot of tremor and it's not getting much better, increasing more toward 200 hertz will tend to make that better. Why? I don't know the theory behind that. Also, we have patients who have sometimes a worsening gait disorder or gait freezing from high frequency stim. If you reduce the frequency some, and some people reduce to 60, 80 hertz, you can preserve at least a moderate amount of benefit, but reduce that freezing side effect. So there are all kinds of interesting, unexplored effects of frequency that probably will give us many, many clues. One that I'm fascinated by is, these gamma band oscillations, that is around 60 to 90 hertz, that will occur, in the brains of people who have been exposed to a lot of dopamine, especially when they're on levodopa. So stimulation can entrain and drive that frequency. And we still think this may be a mechanism behind stimulation-induced dyskinesias, is that you're driving a gamma rhythm, which is normally a prokinetic rhythm that is needed for transiently in a coordinated way for normal movement. It's been studied in jugglers who have cortical gamma oscillations coming in and out during certain phases of juggling. If you drive that rhythm, have it going all the time, it's like you're juggling all the time, right? And that would be one theory for why people become dyskinetic. So, can we tune the stimulation in a way that it does not drive the gamma sort of resonance frequency, or drives it just enough to facilitate normal movement without spilling over into excessive movement? These are all really interesting questions we're just beginning to look at.
So maybe the oldest question for basal ganglion Parkinson's disease is, what can we learn about the organization of the motor system by looking at Parkinson's disease? Because I don't think very many, like, I don't know, robotics engineers would have imagined a system in which something went wrong and you got the symptoms of Parkinson's disease. You know, it doesn't seem like there's just a mismatch of proprioception and dysmetria or something like that. It doesn't seem even like it's not just a failure to initiate movement, because movements once initiated are still running too slow or worse. So is there a bottom line for motor systems people looking at basal ganglia diseases and then trying to figure out what is the information flow that gives rise to normal movements, good movements?
Yeah, a huge debate is, what can you learn about normal function from abnormalities, especially with invasive recording? We're not able to study normal function invasively, only non-invasively. I almost feel that's a philosophical question a bit. You know, in my early days, you know, grant applications would be attacked a lot for just that question. More recently,
Didn't mean to touch that.
More recently, you know, the NIH and federal funding agencies have put a lot more emphasis on human research, especially through the BRAIN initiative, announced through the Obama White House in 2013, which has funded a lot of the kind of work that I've been talking about. So there was very much a bias against human studies before, with that idea, well, we can't figure out normal function from abnormal brains, but that's changed quite a bit. It's a classic thing in neurology that you try to understand something about normal function from abnormal function. You know, for example, as I mentioned earlier today, my colleague Paul Larson found that lesions or stimulation in a part of the caudate nucleus, you know, ameliorate tinnitus. And so what does that mean? Does it mean that that part of the caudate is involved in, you know, phantom perception? And what does that mean for normal perception? So I think there are really fascinating, you know, leads that you can have in this area. But it is true that it's hard to tease that out 100%. You know, I remember, you were probably there. There was one of the meetings of the International Basal Ganglia Society, where there was a panel on what do the basal ganglia do, and there were 4 different experts. This was like 8 or 10 years ago or something like that, and you know, there was a surprising lack of consensus. I remember one answer from one of the Israeli labs was dimensionality reduction is what the basal ganglia do, right? And then of course there were the models of you know, action selection. But, you know, we still don't know very well. There's, of course, enormous interest in motor learning. And one of my colleagues, Doris Wong, is working on that also with our invasive chronic recording patients, and has shown some very interesting theta band changes in the striatum as someone successfully learns a motor sequence, which is exactly what's seen in rats running a maze who are learning a new sequence. And that also brings up something else. You know, I look at human research as very symbiotic with animal model research, right? We might be able to identify certain relevant rhythms or problems, and then you can go to an animal model or a normal animal and test whether that really is causally related to the problem. So by going back and forth between, you know, animal research and human research, that's one way to help address this problem where, well, if it's an abnormal brain, are we learning anything about function?
I suppose at the end of the day, it's enough that you heal people and make them actually able to function.
You know, another, I guess, you mentioned sore spots about grant referees. We all have them. And my work is often accused, kind of correctly, of being not very mechanistic, right? And that's somewhat the nature of human work. We don't have as good a tools as, say, Charlie, you have in your lab to to dissect mechanism. And then I also tend to care a little bit less about it than maybe I should, because at the end of the day, I'm trying to improve a therapy, right? And so if we have these identify brain activity that we can harness to drive a DBS system better, that's kind of a little bit more my interest than figuring out the exact mechanism of it. I have to admit, I'm a little embarrassed to admit that. So that criticism of this work not being that mechanistic is a good one.
Everyone can't do everything.
Well, people who are
right.
studying mechanism have to have something to find out the mechanism of, right? There has to be a phenomenon that matters, that you know you care about. And then you would set out to find the mechanism of that. The worst nightmare for a mechanistic scientist is to find out the mechanism of something that doesn't matter at all, and to spend years doing it.
Yep.
So true.
Yeah, I will mention one. There's been a couple of exciting interactions which sort of illustrate what you're talking about. So a group in Sweden was working on a rat model of dyskinesia and discovered this cortical gamma oscillation in rats that became dyskinetic from chronic, overuse of levodopa. And they were quite excited to find when we found the same thing in dyskinetic humans, and it looked almost identical. It's the one time I've seen rat data and human data look almost identical in a pathological condition. So that was, exciting for both of us to do that. And likewise, my colleague Bob Knight at Berkeley has done a lot of studies of normal cortical function in cognitive neuroscience, looking at phase amplitude interactions and how those mediate different cognitive functions. And then we had a study that found in Parkinson's that the motor cortex has abnormal phase amplitude interactions. So I think that excited Bob Knight to see, wow, it's when you have something you think is normal, and then when it goes wrong in a disease, that's a nice link.
Thank you so much for joining us. I know you have an incredibly busy schedule, and this has been a treat. And thank you, Philip Starr, for joining us, and everyone. This has been Neuroscientists Talk Shop.
Yeah, thanks very much.
Thank you. Yeah, this was great.
Thanks
Sorry
so
about the rough start there with the